Hello, everyone, and welcome to today's keynote presentation, Uncovering Immunological Mechanisms of Protection from Infection and Vaccination in Humans. I am Susie of LabRoots, and I'll be your today's moderator. Now let's get started. Before we begin, I would like to remind everyone that today's event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them in the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box at the far left of your screen. I now present today's speaker, Dr. Marcelo B. Sztein, Professor of Pediatrics, Associate Director for Basic and Translational Research and Immunology Group Leader at the Center for Vaccine Development and Global Health at the University of Maryland. For a complete biography on Dr. Sztein, please visit the Biography tab at the top of your screen. Dr. Sztein, you may now begin your presentation.
Good morning. I would like to start by thanking the people at Fluidigm who has invited me to present some of the work conducted in my group. And I would like to say at the outset that this is a slide presentation that will be covering the mechanisms of protection from infection and vaccination in humans, and the use of mass cytometry that can accelerate the development of vaccines and the understanding of the mechanisms of infection in humans. So to start with a general description of the vaccine development pipeline, the development of vaccines is a very complex enterprise involving many stages, including a discovery phase, an exploratory stage, typically performed in animal models, all the way to a preclinical stage. And those vaccine candidates that are deemed appropriate proceed into the phase 1 and phase 2 studies, which are small human studies, in which many immunological studies are performed in an attempt to identify the immunological correlates of protection that to verify during the preclinical stage that those immune mechanisms are also translatable to humans. And because of the limitations on availability of cells during the phase 1 and 2 clinical studies, particularly in studies conducted in children, and also due to the complexity of the immune response, technologies such as mass cytometry that allows the measurement of many parameters simultaneously can have a great impact. So I will address in this presentation how we have successfully used mass cytometry to help identify the immune responses which might play a role in protection and natural infection, as well as during vaccination. Of the many organisms studied in my group, this talk will be focused on Salmonella Typhi. And the reason why I selected this organism is because it's a disease of great public health importance, which causes about 17 million cases and 200,000 deaths annually. So I will focus on 3 different projects that we performed over the last few years, in which we have used mass cytometry to, for example, identify the key role that T regulatory cells play in the development of typhoid fever by using a unique model in which volunteers are orally challenged with wild-type Salmonella Typhi. I will then move into a series of studies in which the cellular immune responses are measured in both children and adults with the endpoint of comparing whether the responses are similar or not. And I will conclude by a very interesting series of experiments in which we studied the epigenetic modifications elicited in cells isolated from the human gut and exposed in vitro to Salmonella Typhi. These experiments are important because it can have a great impact in the responses at the gut level. So the first part of the study will be that referring to T regulatory cells. And just to give you a basic understanding of a typhoid fever, for those of you who are not familiar, this disease is caused by the bacterium Salmonella Typhi. It's spread via contaminated food and water. The individuals can develop chronic asymptomatic infections which can spread the disease and cause acute infection in others, as well as a big problem related to the emergence of antibiotic resistance. As I mentioned, Salmonella Typhi is a gram-negative, human-restricted facultative intracellular pathogen, which makes the studies in humans very important to understand the pathogenesis as well as vaccine development. This bacterium is transmitted via the fecal-oral route, and after accessing the host, it disseminates with the bloodstream into systemic and sometimes chronic infections. And one of the two vaccines that has been licensed in the United States is the oral light attenuated vaccine strain Ty21a. Now, over the years, we understood the immunogenicity of this vaccine and defined that both antibody and memory B cells are induced. However, they tend not to correlate with the protection from human wild-type challenge infection. Also, we studied and reported that CD4 and CD8 T cell responses appear to play an important role, both in terms of cytokine production as well as contact-dependent cytotoxicity. This diagram is from a review that my group published a few years ago, in which we have developed an understanding of what does the immune response to Salmonella Typhi entail in humans. And they include T memory cells, they include B memory cells, antibody secreting cells, that means the humoral arm of the immune response, dendritic cells, and an important role of chemokines and cytokines in both the activation of antigen-presenting cells, macrophages, dendritic cells, as well as a polymorphonuclear cell activation, and even bystander stimulation and clonal expansion of B and T cells. However, T regulatory cells remain undefined. So, in spite of this complexity of the immune response to Salmonella Typhi, the key question remains unanswered. Which of these cell-mediated immune responses, if any, are associated with protection from typhoid fever? And why is this important? The identification of the precise immunological correlates of protection, either mechanistic or non-mechanistic, can not only help define effector immunity to be pursued during vaccine development, but also help predict long-term protection, as well as accelerate the development of broad-spectrum vaccines to other enteric fevers. For example, Salmonella Typhi A, Salmonella Typhi B, as well as other enteric infections like Shigella or enterotoxigenic E. coli. Now, how can we address this question directly? And one way in which this can be addressed, particularly given that the Salmonella Typhi, as I mentioned before, is a human-restricted pathogen, is to study volunteers who are challenged with wild-type Salmonella Typhi. And for this, we enter into a close collaboration with Andy Pollard in Oxford. And during this human challenge model, of the many parameters that we have measured, we concentrate in trying to identify the correlates of protection. And for the purposes of this talk, I will focus on all of the cell-mediated immune response studies on T regulatory cells. This is the study design. Basically, volunteers were challenged with wild-type Salmonella Typhi, and then at several time points, we obtained cells which were used in the present study. Now, some of these volunteers who were challenged developed typhoid disease, while others did not. And the approach was to compare the immune responses that were present in volunteers who were developed typhoid fever and those volunteers who did not. By comparing and contrasting those responses, we can have a more educated understanding of what are the immune responses in humans that correlate with protection. So, for this talk, as I mentioned, I will concentrate in regulatory T cells, and why these cells are important is because they work as the counterbalancing arm of the immune response by suppressing the response of the effector memory cells. So a dysregulation of the regulatory response can have a strong impact in what the ultimate immune response is generated and whether this response stops being protected because an overregulation by deregulatory cells. So the goals are those that I just mentioned, which are to identify the potential role of circulating regulatory T cells in a wild-type Typhi challenge model in humans, and second, to identify which is the homing protection and activation characteristics of the regulatory cells that are associated with typhoid diagnosis. And here, if you focus where my mouse is indicating, we can see that even before challenge, some individuals have a regulatory response, particularly on regulatory cells that express integrin alpha-4 beta-7, which is a gut-homing molecule, than those volunteers here in red who did not develop a typhoid disease. If we look in panel B, we can see that after challenge, 1 to 4 days in this case, 21 to 28 days in this case, there are lower irregulatory cells in circulation that express alpha-4 beta-7, probably indicating that these cells have home to the gut. Here, we are showing the levels of deregulatory activation in representative volunteers, and as you can see, in those volunteers who develop typhoid disease, those in blue, the cells, the deregulatory cells, are more activated than in those who did not develop typhoid disease, suggesting that the upregulation of a type-specific expression of some markers, particularly near the time of typhoid diagnosis, support an association of increased activation of deregulatory cells and the development of typhoid disease. Now, we went a step further, and we performed depletion studies of irregulatory cells. In this case, we isolated peripheral blood mononuclear cells. Some of them were depleted, and some of them were not depleted of irregulatory cells. And then they were exposed to SIV-infected autologous cells, and after 14 to 18 hours, we stained the cells by mass cytometry, and that allowed us to measure many parameters at the same time, trying to see whether depletion of irregulatory cells have an effect on the T effect of response. And this is what we have seen, that once we deplete irregulatory cells, these are the fold increase of depleted versus toxicity, we can see a significant increase in cells that responded to TNF alpha, as well as those that are double positive. And these increases were observed both in cells expressing alpha 4 beta 7, which is an indication that these cells are coming to the gut, as well as alpha 4 beta 7 negative cells. So, in conclusion from this section of the talk, a type-specific expression of integrin alpha 4 beta 7 is upregulated pre-challenge in those volunteers who develop typhoid disease. A type-specific expression of activation molecules is increased in typhoid disease volunteers, and the T regulatory cells are capable of functionally suppressing a type-specific responses, giving an indication that the responses following challenge with wild-type typhoid on whether the volunteer develop or not typhoid disease points to a role of the regulatory cells. And I would like to acknowledge particularly Monica MacArthur, who performed this study in my group, as well as our collaborators at Oxford, statisticians, Byron Levine with clinical studies at the CBD, and many of the people involved in this important experiment. Now, moving to the second topic, I would like to discuss some of the differences observing the T-cell-mediated immunity elicited by oral immunization with the licensed Ty21a attenuated typhoid fever vaccine, which I described at the outset of this presentation, but comparing the responses in children and adults. So, human pediatric immunology is very important because the responses in children and adults are not necessarily the same. And there is a very poor understanding of the human pediatric immune response. And this is due to many reasons, particularly accessibility of volunteers, the quantities of cells that we can obtain, time points that can be studied, etc., which makes them a critically underexplored population. Now, a mass cytometry is one of those technologies that, because of its cell-sparing characteristics, makes the measurement of multiple parameters possible with a fewer number of cells. And this is particularly important in the study of pediatric populations in which the accessibility to cell numbers is very limited. Regarding the differences in the literature between children and adults, it is clear that lower percentages of CD4 are observed in children. Also, there is an increase in the percentage, lower percentages of naive T cells, and a higher percentage of naive T cells in children, and that younger children are less likely to develop robust immune responses. So what do we know from the field? In one of the few studies in which children of various ages were compared, a study that goes back to the early 80s in children in Chile. Children which are of lower age have a somewhat lower response in terms of a protective efficacy than those children that are 10 to 14 years old or 15-year-old children. And although it's a lot of overlap, there is a trend in efficacy. Here we can see 59% in the 5 to 9-year-old, going to 67% in the 10 to 14, and 85% in the over 15-year-old. Because of this field data, we started this study trying to understand what is the difference among different age strata of children, and which are the differences in the responses between pediatric and adult recipients. And here we have some phenotypic data by looking at CD4 T cells in 6 to 15 years old. This is in blue. 16 to 17 years old. These are in orange, and adults 20 to 65 years old in black. And as you can see in children, as previously reported, we have higher percentages of naive cells and a significantly lower percentages of effector memory cells. And this is the percentages of the memory population. In this case, it is for positive cells. And but this is just a phenotypic characterization. What is important is how do these cells respond to antigenic stimulation. And we use several systems, but I will focus this talk in the use of Staphylococcal enterotoxin B, or SEB, which is a super antigen that, because it cross-links the V beta subunit of the MHC class 2 with the TCR, stimulates a large number of cells. And this robust activation will allow us to compare what you will call the overall T-cell mediated immune responses in children versus adults. If we look at the CD4 levels of T activation as measured by CD69 expression, which is a marker of general activation, you can see a close statistical correlation as the children age. In contrast, in adults, those differences are not apparent. You can see a T of 0.81, showing that throughout age, the responses to SEB do not change in adults. So we identify these age-associated increases in activation potential among CD4 positive cells in children. If we look at CD8 responses, we see very much the same phenomenon. The levels of activation increase as the children age. However, in adults, the response is flat. Also, extremely important is that we study T-cell multifunctionality, that is the ability of cells to produce more than one function, for example, interferon gamma and TNF-alpha production together with IL-17 or other functions. And what we can see is that as the children age, there is really not much difference when you look at monofunctional T-cells. However, there were very strong correlations, statistically significant, when we look at 2 functions, 3 functions, or more than 3 functions in children, indicating that the children of a younger age seem to have a lower multifunctional responses than those at let's say, over 15 years old. If we look at CD8 positive T effect on memory cell multifunctionality, we see the same response. One function just barely makes it to significant, but two functions, three functions, and more than three functions are highly significant and increase with age. Again, indicating that the functionality of these CD8 positive T effector memory cells are strongly correlated with age through adolescence, in this case, after stimulation with the super antigen. And this is important, they reach adult levels by the teenage years. So in summary of the SEB response, we can see that children have decreased activation, decreased responses. I didn't have to show you the individual data, but this published in this paper in Frontiers in Immunology in 2018, in which many cytokines are produced at lower levels in children, and particularly the multifunctionality is lower in children than in adults. And pretty much the same effects over age are observed with regard to the CD4 positive cells. And this is what I just mentioned, that 16 and 17-year-old T cell activation and function are already more adult-like. But one thing is to study super antigen response as a general stimulation, but the much more important thing is to study the specific immune response in children who have been vaccinated with Ty21a by studying the autologous Salmonella Typhi stimulation response. And we use a protocol similar to what I showed before. We have the target cells, which we gamma irradiate and left uninfected, compared to target cells that follow the same procedure, but they have been infected with Salmonella Typhi. And then we combine them with BNC obtained from the children. And what we have seen is that for CD4 cells, we see increases in the responses that are significant pre versus post-vaccination in both children, as indicated by the blue or orange symbol, as well as adults. In contrast, in CD8 positive cells, we can see the increases that we typically observe in adults, but in children, in part due to a great variability, did not reach statistically significant levels. But to study in a further depth these responses, since we are working with mass cytometry with over 30 parameters, we use a ESNE, which is one of the classical dimensionality reduction tools, to study the variation between children and adults. And the advantage of this is that this provides an unbiased means of identifying key population features that will be very difficult to discern using conventional gating methods. And here I included an example of a ESNE map, and you can see in children, which are the two top quads, as well as adults in the two lower panels. If you look, for example, at cluster 12, it's very easy to see that they have less of a cell representation than adults. And the same thing for cluster 5. When we analyze this data, for example, for CD8 positive, CD69 positive, that is activated cells, we can see that some of the populations are higher in children, for example, here with the blue bars, than in adults. For other clusters, adults are have a higher representation in the clusters than children, and we can see that many clusters, including 7, 12, and 15, and 22, In adults, you see a higher representation in those clusters. Now, this is not very relevant unless you know what is in those clusters. So we use gating out of those clusters to identify which are the characteristics in each of those clusters. And here we have clusters 1 and 5, which are overrepresented in children, and the rest of the clusters, which are overrepresented in adults. And as you can see, just by looking at the proportions of cells that produce transient P or interferon gamma or IL-2, that in adults, the response is more multifunctional than that observed in children. And this is probably better exemplified on this panel, on the right panel, in which cells exhibiting more than 3 functions in black or 3 functions in dark gray, they are really concentrated in those clusters that are overrepresented in adults. And we can also, we did study homing molecules, other non-cytotoxic 1 cytokines, other molecules that are important in cell activation, all concomitantly. So we could characterize in great detail the different clusters and which are their levels of functionality. So to summarize this autologous part of the presentation, in which cells were specifically stimulated with an antigen, again, we see that children have decreased baseline multifunctionality as compared to adults. The adults have an enhanced activation following immunization with the Typhoid vaccine Ty21a. And very importantly, in children, there are fewer cells that exhibit multifunctional properties. And this basically summarizes these important findings comparing children and adults, of which less multifunctionality appears to be a defining characteristic. In contrast, adults tend to have higher levels of activation. And although the responses to Ty21a vaccination is similar in terms of the overall response, there are important differences in the responsiveness at baseline. And for this section of the talk, I would like to acknowledge Mark Rudolph, an ex-PhD student in my group, as well as Monica MacArthur, who performed the majority of this experiment. And now we'll go into the third point of discussion during this presentation, and this has to deal with the early cell type specific epigenetic modification elicited in cells which were isolated from the human gut following ex vivo exposure to Salmonella Typhi. So these are the results of a paper that was just published last month in Scientific Reports. So this data is already available for you to look in detail. So why to study epigenetic modifications? And these are the main reasons. The epigenetic profiles that are elicited upon stimulation of cells are key molecular processes which underlie the differentiation of cells throughout the maturation process, all the way from progenitors to functionally mature cells. There are many epigenetic modifications, including acetylation, methylation, phosphorylation, etc. And these modifications dictate chromatin structure and function and regulate gene expression. And in fact, certain histone epigenetic modifications have been associated with human disease. Now, the bacteria in these epigenetic changes are simply very little to no reports that have studied them in detail in humans, particularly at the level of the tissues, in this case, the human gut. What is important to keep in mind is that the alterations in the chromatin marks regulate the gene expression that can lead to either suppression or activation depending on the particular epigenetic modification. And the hypothesis of the present study was like that the exposure to bacterial organisms will result in specific long-lasting impact on the home responses and therefore to bacterial infection. Here I borrow a graphic from the beautiful review that Kali Fabregat and collaborators published earlier this year in Genes, and they point that at all levels of differentiation and activation, there is an increasing numbers of epigenetic modifications that have been described and published. And they include all cell lineages, the lymphocytes, CD4 helper cells, CD8 cytotoxic cells, NK, granulocytes, macrophages, dendritic cells, and taken all together, this points to the importance of epigenetic changes in the immune response. So, to reiterate, the rationale for studying this epigenetic modification in SIV infection, terminal ileum in humans is the main segment of the GI tract in which the pathology associated with SIV infection has been reported and is considered the favored site of infection for Salmonella Typhi. The vast majority of immunological studies performed in humans involve by necessity the study of immune cells in circulation, and very little is known about the mucosal immune responses at the level of the gut. And my group have been studying this for several years, and we have reported a lot of these cell-mediated immune responses in the human terminal ileum following Ty21a immunization. However, no information is available on the early epigenetic signature elicited in cells isolated from the human terminal ileum following ex vivo exposure to wild-type Salmonella Typhi. But since changes in the epigenetic landscape are likely to markedly regulate the local immune response, we hypothesize that the study of epigenetic modification is going to be one of the key factors to be considered. So how did we study epigenetic changes? And we used a tool called Epitome, which is basically a tool to understand the epigenetic landscape profile that was originally reported by Chung et al. In Cell in 2017 to describe the epigenetic changes in aging. And then that was followed by another publication in 2018. And the advantage is that it's a mass cytometry-based analytical platform that simultaneously measures epigenetics and immunological markers at the single cell level. And this is based on the availability of monoclonal antibodies that can study many of the epigenetic modifications, including acetylation, methylation, phosphorylation, etc., as well as non-covalent modifications, including histone variants. And as you can see, there are monoclonals that recognize these epigenetic changes in all of the histones, histone 1, 2A, 2B, histone 3, and histone 4, and they involve both methylation and acetylation, and all of the other signatures I just described, which are very clearly illustrated in this diagram from the Clinical Immunology 2018 paper by Cheung et al. So the overall strategy is to take advantage of epitope to create a cell epigenetic atlas in cells isolated from the human terminal ileum following in vitro exposure to wild-type Salmonella Typhi. By using this high-dimensional analysis, using ESNE, Chromosome, and other modern tools, and some other even more advanced tools developed by Astrolabe, although these were not used in the present studies, we are collaborating with them in trying to understand this phenomenon in further detail. We compare and contrast the chromatin modification between and within the different cell types following exposure to Typhi. And this is a critical issue. Because it's mass cytometry, we were able to collect 8 to 10 million individual cells for each volunteer in each experiment to assure that sufficient number of events could be analyzed for each of the cell subsets and for each of the epigenetic modifications. So the experimental design of this study involves 2 stages. The first stage was the isolation and cryopreservation of epithelial and cells. For this, the tissues from the terminal ileum were disaggregated and injected with collagen spores based on DNA. We generated distinctive cell suspensions, which were filtered and cryopreserved in liquid nitrogen until we were ready to run the experiments. On the day of infection, we store the cells, we incubate them for 2 hours, and then we expose them or not to wild-type Salmonella Typhi. And this allowed us to compare directly those epigenetic modifications in the presence or absence of Salmonella Typhi. And after the incubation, we used a panel of 33 metal-labeled monoclonal antibodies and analyzed the data by mass cytometry. Overall, it's a panel of 33 metals that include cells, multiple subsets. In fact, we tested 11 major cell subsets, including B cells, CD3 positive T cells, CD4, CD8, NK, TCR gamma delta, MATE, which are the mucosal associated invariant T cells, NKTs, monocytes, macrophages, and epithelial cells. So mass cytometry allowed us to study all of these subsets concomitantly with the expression of 6 classes of histone modifications and 1 histone variant. And here I am showing the supervised clustering of epigenetic changes between and within the different mucosal cell subsets after Typhi infection. And you can see how media and TY2 have different ESNE maps, and that each cell subset, in this case CD3, in this case CD4, in this case CD8, in this case epithelial cells, have very different ESNE maps. So we color coded the location of the epigenetic marked metaclusters, and this is what we see here, so we could study these populations in further detail. In this slide, I am showing the supervised clustering of the epigenetic changes associated with exposure to Salmonella Typhi. And from the 225 clusters that we identified, the data was grouped into 15 meta-clusters and organized based on their similarities in high-dimensional space. And these studies were performed using the Chromosome algorithm and Cytometry. And these are, of course, identification in trees with nodes at the same sites. Now, when we look at the chromatin changes elicited by Salmonella infection, we can see that in certain epigenetic marks, we see increased expression. For example, in this case, acetylation of the lysine in the H2B histone, and so on. We have several that have increased expression and several of the epigenetic marks in which the difference between stage infected and non-infected actually was negative. So when we look at the meta-cluster maps of the mucosal subset abundances, and this is for all 11 subsets that we studied, we can see in the pie chart that those that were incubated with media and those that were incubated with EY2 were clearly different in terms of the epigenetic modification. But you can look at this area and compare in the presence of Salmonella Typhi versus the same area in the absence of Salmonella Typhi. And we are continuing these studies by looking into further detail at these differences in the absence or the presence of Salmonella Typhi. Now, the other advantage of mass cytometry is that it allows us to study the multifunctional patterns of epigenetic changes in the various mucosal cells. And as you can see, there was a very significant difference between the chromatin markers that are decreased and those that are increased in the presence of Salmonella Typhi. But since we have all of the labels in the same queue, we could gauge on, for example, CD4 positive T cells in this slide, and we can see that there is a significant increase in arginine methylation in CD4 positive cells following exposure to Salmonella Typhi. Also, we could dig into further detail by looking at CD4 positive cells, which have overexpression of the arginine methylation, position 2, and we associate these changes in these cells with functionality. And in this case, in the left panel, we can see that the T effector memory cells are significantly overrepresented in cells that are gaining on a basis in arginine methylation 2, CD4 positive T cells. And interestingly enough, they are significantly decreased for the expression of CD57, which is a very important maturation marker. If we go one step further in this multifunctional analysis, and we do hierarchical clustering of CD4 positive cells, this was done using principal component analysis, we can see when we group by media or exposure to Salmonella Typhi, different increases, that arginine methylation is a major driver, as we discussed before, but arginine methylation sometimes is combined in certain subsets with other epigenetic marks that are overexpressed or underexpressed simultaneously. And this is a story that is just starting, but it's a lead into further in-depth study of the epigenetic landscape. So, in conclusion, Staph infection does indeed result in epigenetic modifications that are cell type and subset specific. These include multiple epigenetic modifications, including the methylation of histone 3 and lysine 4, histone 4 and lysine 20, acetylation in some cases, and arginine methylation as well. These epigenetic marks have already been shown to affect differentiation in other systems, so we are presuming that they are likely to also have a major effect in T-cell differentiation and gene expression following exposure to Salmonella Typhi. And this might be important, among other things, because, for example, arginine methylation controls the strength of the cell signaling via the gamma chain family of cytokines, which include IL-2, IL-4, IL-7, IL-15, IL-21, etc. So the overall conclusion is that understanding the action of the epigenetic regulator, which determines the fine gene transcription pattern in different cell types and subsets, is essential in characterizing the pathways associated with protection from infection, as well as those that will result in the development of robust and effective host immune responses. These studies, as far as we know, are the first proof of principle that these epigenetic changes in mucosal cells isolated from the human terminal ileum can be directly linked to exposure to a gut pathogen, which is a major source of disease. And future studies are needed to really further dig into these responses, establish temporal patterns, and how these results in the differences in the development of protective immunity. And I would like to acknowledge Rosangela Nesgani, who performed these studies, assisted by Diane Chen or Regina Harley, as well as Andrea Bafford, the surgeon, who provided us with the terminal ileum tissues, of course, the cells of support. And with that, I would like to thank you very much for your attention.
Thank you, Dr. Sztein, for your informative presentation. We'll now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click the Ask a Question box located at the far left of your screen. Let's get started. Our first question is, why is it important to study the difference between immunity in children and adults?
Well, as most of the audience knows, children respond differently than adults. And in a case in which we are studying responses to vaccination, it's very important to know what are the differences between the immune responses in the children than in adults, because that can markedly influence not only the effectiveness of a vaccine, but also can have marked effects on the duration and the quality of the response. The reason why I brought the example of children versus adults is because by studying those differences, hopefully we can understand whether the mechanisms of protection are the same in children and in adults, and what are the specific determinants on the characteristics of the populations that we can study to define how the vaccines are expected to work in children versus adults, or the elderly for that matter, because there are many associated differences with aging that are also very important for vaccines that are directed to the elderly population.
Awesome. Just a follow-up question to that is, there have been several efforts to obtain global immune maps of healthy immune systems in adults. Is there any such effort underway for children that you're aware of?
Unfortunately, and I might be wrong, but I am not aware of any similar efforts that are underway in children. Definitely, it's a very good question and something that should be explored, given the importance of the relative differences between children, adults, and the elderly. And I will say probably among all age strata.
Thank you. And it looks like we have a couple questions coming in from the audience. Thank you. One of them is the epigenetic changes been looked at in functional assays?
No, but that's another excellent question, and that is what is coming next. And the reason is, that's why I mentioned at the outset of that component of the presentation, that this is almost like a proof of principle in the infectious diseases field. A lot of work remains to be done and studying at the functional implications of the epigenetic modifications that we have seen after exposing the cells to wild-type Salmonella Typhi is very high on the agenda.
Thank you. And it looks like we have another question from the audience here. Can you comment on the effect of the adjuvants, excuse me if I mispronounced that, in a vaccine in adults versus children? Does that account for these differences to some level?
Well, these are all very, very good questions. The question of the adjuvants is particularly important because some of the proposed mechanisms of adjuvanticity is through a regulation at the level of epigenetics. So to my knowledge, that was never studied in humans with cells isolated from tissues, like, for example, terminal ileum, as the data I presented. So that's definitively an avenue that has to be explored in the adjuvant field. Thank you for that question.
Great, thank you. Off the topic of children, but just going into cytometry, are there any other examples in humans other than Salmonella Typhi, which in mass cytometry has been used to study human challenges?
Yes, I didn't go into some of those performed in my group because of a lack of time, but we have studied a model in which enterotoxigenic E. coli wild type was used to challenge volunteers, and in those studies we also studied the responses using mass cytometry. And as a matter of fact, we identified very interesting correlations between T follicular helper cells and their expression of interleukin alpha-4 beta-7, which is a mucosal homing markers, and for example, the level of diarrhea that in terms of volume in volunteers who have been challenged with ETEC, enterotoxigenic E. coli, some of whom were protected and some of whom were not. So there are other infections in which this can be studied, and I think it's a very, very important avenue to pursue, given the fact that in humans, challenge models are as close as we can get to real life.
Thank you. And it looks like we have about 1 minute left, so I'm just going to ask one more question here. Do you envision EPITOPH to become a valuable application for CyTOF users? And if so, what is needed to make this a more accessible application for CyTOF users?
I think that it is going to be very important for CyTOF users. And the reason I say that is that this is a technology that can allow the concomitant measurements of many, many epigenetic modifications in many cell subsets at the same time. But instead of collecting just a few hundred cells because of cost, for example, in these studies, we collected about 8 or 10 million cells per sample. So the data that we obtain and that other groups might obtain They are in millions of individual cells, measuring, of course, up to 50 plus parameters. So my expectation is as time goes by, more research group will look into this technique as a possible forefront application of mass cytometry. I guess only time will tell.
Yeah, right. Well, thank you so much for your time today and so much for all your important research. We really appreciate hearing from you. Before we go, I'd like to thank the audience for joining us today, and questions submitted during the on-demand period will be addressed by the speaker via contact information provided at the time of registration. This webcast can be viewed on demand. Just give it a day or so, and you can review the presentation here today. Until next time, goodbye.
Thank you very much.